Okay, hey Biochem, so this is the last metabolic regulation video, and it's probably going to be one that you might need to watch a couple times or be patient with yourself. It's not one of those concepts that your brain just kind of easily soaks up. You kind of need to let it marinate for a while, so be patient with yourself and maybe take a break, rewatch it, and come ask questions if you need help. Okay, so these are the pathways that we're going to do. You should be able to describe the regulation for each of the following pathways: glycolysis, gluconeogenesis, PDC, and TCA, and combined these are called carbohydrate metabolism, because that's carbohydrate catabolism and carbohydrate anabolism. And then fatty acid catabolism, which includes these three processes combined together, and then fatty acid synthesis, which is, I guess I should have written it out, acetyl CoA carboxylase plus fatty acid synthase. Okay, and we're going to focus on, maybe I'll just talk just a little bit to give you something to think about. So we're going to focus mainly on the irreversible enzymes, or the steps that catalyze the enzymes that catalyze the irreversible steps. And in some cases, we're even going to focus very specifically on the rate limiting enzyme. And before we get into the nitty-gritties, I'm going to draw how I think about this in my mind, which is an analogy. So all of these pathways, it's not either directly on or off. It's more of like a flow through or a flux, where sometimes there's a little bit more of the intermediates going through than less. So I haven't actually drawn this out before. So if you think about, let's just say, a pipe that water runs through, okay, going through like this, and the pipe would be a pathway, like let's say glycolysis. So really it's not, okay, so it's not water running through, it's, I still can't remember where that eraser button is here. So it'd be glucose, whoops, I was using yellow. Oh. Yellow. Glucose going in and pyruvate coming out, and there's just a constant like flow of these intermediates going in and out. And again, we're not thinking about like, this is just the carbon intermediates. Well, the irreversible steps, I think about them as, hey, I need to be able to erase. I think about them as like places where the pipe gets really thin or scrunches up. So like the first step in glycolysis with hexokinase, like the irreversible step makes it so that if it's tightly regulated at this spot, it really affects what goes in and what goes out. And the most regulated step in each pathway is the rate limiting step. And the reason for this, so let's do, oh no, the fluffs are upset. That's the golden doodle, in case you were wondering. I wonder if I just ignore her, if she'll go away. So the irreversible step that catalyzes the rate limiting step in glycolysis is catalyzed by phosphofructokinase, PFK. And the reason that this is the most tightly regulated is because if you imagine that the pipe kind of gets thin here, but there's a valve, I don't know how to do this. Like, think of like when you turn your sink on or your sink off, and you've got one of those valves where you're able to rotate it, where it's either on in one direction and off in the other direction, so it like gets squeezed tighter or the flow opens up. That's the regulation, is this sort of nozzle that you're able to open up the pipeline a little bit more or squeeze the pipeline tighter. And if you squeeze the pipeline tighter and it gets smaller, then you'll get less flow through coming out. So I don't know if that makes any sense, but that's how I think about regulation, is that all these pathways are like a pipeline and there are intermediates flowing through, and the way that you can control the amount that comes out of the pipe is to either like squeeze the pipe closed or somewhat closed at these irreversible steps, or open the pipeline so that more comes out. So that's like the cartoon idea of how I think about this. Okay, so we're going to start with carbohydrate catabolism. And this image right here just shows that your blood glucose range has to be maintained in a very narrow range. So the normal range for your blood glucose is right here, and any like major fluctuations beyond this where it starts to dip causes a lot of problems. And so the way to regulate how much blood glucose you have is that you regulate glycolysis and gluconeogenesis. So that's why we're going to start with carbohydrate catabolism. So this is how we get our blood glucose continuously regulated so that you're not pulling, you're not dipping too low or going too high with your blood glucose levels. Okay, so one way that pathways that go in opposite directions, like glycolysis and gluconeogenesis, are regulated is that they're actually in physically different parts of the cell. So physical separation is one mode of regulation that wouldn't have been in the previous video because it didn't quite fit in. But if you see in this figure right here, this is the membrane, or one of the membranes that separates the mitochondria from the cytosol, and part of gluconeogenesis happens in the mitochondria, part of it happens in the cytosol, but glycolysis only happens in the cytosol. So having a physical barrier, a membrane in between the two pathways, is one way of regulating them. Another really good example of this is beta oxidation and fatty acid synthase, where beta oxidation only happens in the matrix, the mitochondrial matrix, and fatty acid synthase only happens in the cytosol, and these are opposite pathways of each other, so they are physically separated in the cell. So that's one way of separating them. So now we're going to look at specific enzymes. We're going to do 2 for glycolysis and gluconeogenesis, and then PDC, the irreversibles, and TCA, and then we'll move to fatty acid metabolism. Okay, so the first irreversible step, and I don't know why I looked through these and they all didn't look pixelated before I uploaded them, but they're a little pixelated now. But I think that you can still see them. So when we look at some of these diagrams, they usually show both pathways at the same time. So what you're seeing over here on the left going down is glycolysis, and what you're seeing on the right going up is gluconeogenesis. So the first irreversible in glycolysis is hexokinase, and the last irreversible in gluconeogenesis is glucose 6-phosphatase, and they're actually reciprocally regulated, meaning one thing turns one off and the same thing turns the other one off. So hexokinase is allosterically inhibited by G6P. Okay, and that makes this product inhibition. Okay, whereas glucose 6-phosphatase is actually allosterically activated by G6P. So not only is G6P the substrate for glucose 6-phosphatase, but G6P can also bind to an other site other than the active site to allosterically upregulate this. And then what we'll talk about, or the ways that we talk about these things, is that it's not that you have all glucose in the cell or all G6P in the cell. You always have a little bit of both of them. And so we talk about them in terms of their ratio. So if we have a ratio of concentration of glucose to glucose 6-phosphate, gotta add the P, and that's less than 1, I call this a high energy condition. Okay, so what does that mean? That means that we have a lot more of G6P, okay, than we do of glucose. When you have a lot more of G6P, that means that glycolysis has been running for too long. So it needs to be slowed down. And also, in addition to that, you probably have a buildup of this high energy stuff, and so you'll move into storage of energy. And that's also represented in this diagram right here, where this piece right here shows that G6P inhibits hexokinase, and G6P activates glucose 6-phosphatase. And so the opposite of this is the low energy condition. So if you have all of these sort of memorized in one direction, you have them memorized in the opposite. So if we have, again, glucose to glucose 6-phosphate, and that is greater than 1, this is considered a low energy condition. Dang it. Okay. So this one is probably the easiest example of all the regulation ones that we'll talk about, and that's why we're going to start with it. And now we're going to go into the rate limiting step of glycolysis and gluconeogenesis. And that's catalyzed by phosphofructokinase. And here I've got phosphofructokinase 1 because there's actually a few different kinds of phosphofructokinase. We'll only talk about phosphofructokinase 1. And this catalyzes the step from F6P to F16BP. And hopefully you remember that this takes ATP and moves that phosphate from ATP onto the fructose to give you ADP. Now this is the rate limiting step, okay, so it's under the most tightest, or the under most tightest, those aren't words that work together. It's under the tightest control because the rate limiting step is the place that you can get the most, let's call it the most fine-tuned control of the output or the flux of the entire pathway. So again, another equation example of things that turn this step on versus turn this step off. And one of the things that I want to point out here is that while ATP is a substrate, at high concentrations of ATP, this molecule will allosterically inhibit PFK. Okay, and the reason for that, again, is if we think about our ratios, again, we don't have all ATP or no ATP. We have a ratio of ATP, and I'm going to put ADP / AMP, because sometimes when we take the phosphate off, we get an AMP versus an ADP. So if you have a lot of ATP, then this will be greater than 1, and that's your high energy condition. And in this case, again, that means that glycolysis has been running for too long. Okay, and will allosterically inhibit. So what I have right here is an image of phosphofructokinase, and the active site here, where the substrate ATP binds and F6P bind at low concentrations of them, will move forward to create F16BP and ADP. However, at high concentrations of ATP, it also binds up here, which allosterically inhibits the reaction and therefore slows it down. So I wanted to give you a diagram so you can see, well, while ATP, yes, is a substrate, and at low concentrations, so this would be where ATP, ADP is less than 1, you'll have your normal reaction, whereas if you increase that ATP concentration, it will come back, and in this case, ATP is a feedback inhibitor. Okay, so why is it a feedback inhibitor? If you remember, if we think about glycolysis, we go glucose, all the way down to PEP and pyruvate. Well, one of the final products of this is ATP, and if you, of that particular reaction, and there's another reaction in here that gives you ATP that comes after F6P, but if you're on glycolysis for too long, the concentration of one of these, this final product of the pathway will come back up to the rate limiting step and feedback inhibit it. So ATP in the case of glycolysis is a feedback inhibitor. There's a few other things on this diagram. So I just want you to know that we will not be covering F26BP. I will also not be covering citrate. So you're only responsible for knowing ATP's regulation, ATP/ADP regulation of F16BP. Or I'm sorry, F1, PFK, phosphofructokinase, this particular step. Okay, and then here's a diagram. You've seen these before. This is a Michaelis-Menten, a quasi-Michaelis-Menten plot that's just showing that at low ATP concentrations, PFK just works as normal. You've seen this before, where the curve is hyperbolic. So you might think about this as something that you've seen with hemoglobin and myoglobin, whereas at high ATP concentrations, it actually reduces the enzyme activity quite a lot. So high concentrations of ATP are inhibitory. I mean, it doesn't completely shut it off, right? We're not completely on or completely off here, but the high concentrations of ATP reduce the amount of F6P that gets used, and also the enzyme activity decreases as well. Cool, huh? All right, moving on. Okay, so regulation of fructose 1,6-bisphosphatase and F16BP. So this is showing both of them. Again, you have glycolysis going down on this side and gluconeogenesis going up over here. And the way that you get glycolysis to run is that you have a high concentration of AMP and you have low concentration of ATP that will regulate, that will positively, these right here, positively regulate phosphofructokinase. Why can't I say that word today? And then if you want gluconeogenesis to run, you need low AMP. And you'll notice that that's actually the only one of these regulators that will affect fructose 1,6-bisphosphatase, while a whole load of things affect PFK. So these pathways are not identically regulated. They're not identical opposites, so they're not identically regulated either. Okay, again, here's a summary diagram of the pieces that you need to know. I guess I should probably cross out citrate on this, although I wonder if you could figure this one out. So citrate, high concentrations of citrate will turn glycolysis off. Where do you find citrate in the cytosol? Well, you find high concentrations of citrate in the cytosol when you're making a fat. Okay, so that's a high energy signal. That means that, again, glycolysis, TCA, ETC has been running for too long and things need to slow down and turn on to storage. But anyways, you don't need to know citrate in the context of this. Just G6P and ATP and AMP/ADP. All right, moving on, PDC and TCA cycle. So the PDC is just an enzyme on its own, and it's regulated mostly by product and feedback inhibition. Okay, so the conditions where the PDC is turned on is when you have a low energy condition. I'm just going to put ADP here. Okay, so when you have a low energy condition, it means you have a lot of ADP, so this will be less than 1. And we're going to introduce NADH here as well, so NADH over NAD plus. And this again, these are concentration things, so this is a ratio of the two. And again, these are turned on in low energy conditions, so again, this is going to be -1 because you have NAD plus and ADP are low energy conditions. And basically the opposites of these are true except for one extra piece. So turned off when you have a lot of ATP, this is your feedback inhibition. And I can show you this on the diagram. And then NADH, you have a lot of NADH, that will come back and product inhibit. Very consistent with these brackets, am I? And then the added one here is that you will also be turned off in high concentrations of acetyl-CoA. Okay, so product inhibition, that's the acetyl-CoA and this NADH. So if you notice in this diagram right here, acetyl-CoA is a product of the PDC, and when you have high concentrations of acetyl-CoA, it product inhibits. And then NADH is also a product of the PDC, and that can, high concentrations of that will also come back and inhibit. Feedback inhibition, that's, is this ATP? Wow, that got really messy. So I don't know if you can see this, but we've got the PDC here, and that connects to the TCA, and this electron thing to ATP, that's electron transport chain to ATP synthase. And so this is oxidative phosphorylation, and again, that would be the very final product of all of these metabolic, catabolic pathways go to ETC and ATP synthase. So ATP synthase, which creates ATP, would be the ATP is a feedback inhibitor because it's something that's made at the very, 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 very end, and high concentrations of that will come back and inhibit the PDC. Sorry, in this case, they've called it PDH. This is also PDC. Okay, TCA cycle. Again, this is the PDC is up here as kind of a recap of the things that inhibit the PDC. What I want you to notice about the TCA cycle is that we're only looking at the 3 irreversible steps right here, and I've tried to white box out some of the things that you don't need to know. But you'll notice that NADH turns all of the irreversible steps off. So again, if we have, let's go to the high energy signals. When we have a lot of NADH, that means you've been running the cycle for too long and needs to be turned off. And let's see, one of these also is regulated by, so this one right here is your rate limiting step, and that's also regulated by ATP. So that is a high energy signal. When you have more ATP, then you do ADP or PI greater than 1, and that will turn everything off. And then the opposites, what turns them on are your low energy signals, and those are just the opposites of these. So I'll just put less than 1 and less than 1 here. So ditto, ditto, opposite dittos. So when your NAD plus is greater than your NADH, this ratio will be less than 1, and when your ADP is less or is more than ATP, this ratio will be also less than 1. And again, this is at all the irreversible steps. And these are NADH is also product inhibition for these 2 steps. It would be feedback inhibition at this step because citrate synthase doesn't make NADH. So this is going to be feedback. And then this ATP is also feedback inhibition, whereas all the other NADHs are product inhibition. And that is carbohydrate catabolism. Are you ready for fatty acid catabolism and anabolism? It's just a little bit different. The way that it gets talked about is actually quite a bit different than the way the carbohydrate catabolism and anabolism is talked about. So these are the different types of regulation that we're going to meet in lipid metabolism. So we're going to have competitive inhibition, some allosteric activation and inhibition, feedback, and then covalent modification. So this is the post-translational phosphorylation and dephosphorylation. So we're going to meet all of these, and we're going to have as many enzymes to look at, though. So that's a good thing. Okay, so the fatty acyl-CoA that comes into the cytosol has two fates. It can either enter the matrix and if it gets entered into the matrix, it will absolutely undergo beta oxidation. There's no coming back. If the fatty acid gets shuttled over into the matrix, right there, it's not coming back out, and it will be broken down for beta oxidation and then to the TCA cycle. The other fate that it has if it gets into the cytosol is that it can just be packaged back into a tag, or it can be made into phospholipid. And the major site of regulation is the carnitine shuttle, and the reason for that is because the carnitine shuttle is the rate limiting step for fatty acid catabolism. And yeah, the major reason for that is just because this is literally sort of like the gatekeeper between if it stays in the cytosol or goes into the matrix. So what regulates the carnitine shuttle is malonyl-CoA. All right. So if you remember from our fatty acid synthesis, you normally find mal-CoA over here after it's been, acetyl-CoA to malonyl-CoA, and the malonyl-CoA goes into fatty acid synthesis. So if you have high concentrations of malonyl-CoA, that is a signal that says you do not need to break down a fat. There is energy, too much energy, and there's energy for storing. Okay, so high concentrations of malonyl-CoA will inhibit that enzyme right there, the one that creates acylcarnitine. And my best guess at this is that this is competitive inhibition. Okay, and my guess for this is because malonyl-CoA is going to look very similar to acyl-CoA. They'll both still have the CoA on them, right? So that's the same, and that CoA group is humongous. Malonyl-CoA has the 3 reduced carbons, or well, actually 2 reduced carbons and 1 carbonyl. And this acyl-CoA is somewhere between 6 to 12 carbons long. So my best guess is that high concentrations of malonyl-CoA, well, I know the high concentrations of malonyl-CoA inhibit that step, and my prediction is that that would be competitive inhibition. I wasn't actually able to find an exact answer to that. We just know that malonyl-CoA inhibits the carnitine shuttle at that first enzyme, which some of you have been asking me, but this is why it's called CPT1. And this one over here that remakes it is called CPT2. Anyways, so high concentrations of malonyl-CoA will inhibit the carnitine shuttle. And the reason for that is if you have lots of malonyl-CoA, it means that you've got tons of energy, enough energy to make a fat, so you better not break the fat. So the acylcarnitine, any acyl-CoAs will not actually transport over into the matrix. And then, of course, if you flip that and you have low malonyl-CoA, it's just the opposite. Okay, and then the last enzyme, or no, we're not the last enzyme, we still have to do beta oxidation. The other enzyme we're going to look at is acetyl-CoA carboxylase, because it's the rate-limiting step of fatty acid synthesis. And there's 2 ways that you can slow this one down. One of them is through feedback inhibition when you have high concentrations of palmitoyl-CoA or palmitate, right? So palmitate, the 16:0 fatty acid with a CoA on it would be palmitoyl-CoA, is the final product after fatty acid synthase is done. And so if you've made a lot of those, then it's a signal to acetyl-CoA carboxylase to be like, okay, we're done making fat. We've got plenty of it. Time to shut down. So feedback inhibition is one of them. And then the second one is this one right here, which is to trigger phosphorylation or dephosphorylation of acetyl-CoA carboxylase, and we're going to talk about that in the next slide. We will not in this class get to the hormone level regulation. If you are able to take Biochem 2, we will get to the hormone level regulation, but right now we're just doing cellular level regulation, not global regulation. So let's talk about the phosphorylation. So the two pieces are feedback inhibition and phosphorylation and dephosphorylation, post-translational modification. Okay, so covalent modification using this enzyme called AMPK, which stands for AMP, which is adenosine monophosphate kinase. That's what AMPK stands for. Okay, so during times of muscle contraction when you need a lot of ATP or you need to run beta oxidation, you'll shut down fatty acid synthesis. And the way that this works is that you have a low energy signal, so our concentration of ATP is low compared to our concentration of AMP, right? So you have a lot of this, which makes a bigger number on the bottom, so this is less than 1, and this will activate AMPK. Okay, so AMP means AMP kinase, so you need a lot of AMP to do this. So in this figure, there's a lot of AMP, and the AMP kinase will phosphorylate acetyl-CoA carboxylase, and that makes it inactive. Okay, so phosphorylated acetyl-CoA carboxylase makes it inactive. Your concentration of malonyl-CoA goes down, which means that the carnitine shuttle is no longer inhibited. Wow. Not inhibited. So the acyl-CoAs get transported across into the matrix and undergo beta oxidation, which eventually gives a whole bunch of electrons to run oxidative phosphorylation, which of course gives you ATP, and that's how you get your ATP from muscle contraction. And so the opposite of all of this is also then true. So if you have high concentrations of ATP, then AMPK, you're going to have a dephosphorylation event where malonyl-CoA, or where acetyl-CoA carboxylase is now active, and then malonyl-CoA concentrations rise, shutting down the carnitine shuttle and shutting down the production of ATP. Okay, so upregulating fatty acid synthesis. Again, we just already talked about one of these, but basically if you have high ATP concentration to AMP, this is greater than 1, then acetyl-CoA carboxylase, this will be dephosphorylated and therefore it will be active. So this is your example of post-translational modification. Other ways to upregulate this is citrate is an allosteric activator. So citrate, then you find it in the cytosol right before it becomes acetyl-CoA. If you have high concentrations of citrate, it can be sort of like a feed-forward activator. So citrate is an allosteric activator of acetyl-CoA carboxylase. Kind of cool, huh? And I feel like I'm missing one. Oh, right. Also, high concentrations of acetyl-CoA also activate. That should make sense because it's its substrate. Okay, and last but not least is oxidation. So beta oxidation, this one's fairly straightforward. So this step right here in beta oxidation that makes NADH will be inhibited by high concentration of NADH. So NADH to NAD plus. When that is greater than 1, that is product inhibition. Okay, and only at that step. And then our second product inhibition also comes at this step right here where we have high concentrations of, will inhibit the thiolase enzyme, and again, that's also product inhibition. Okay, so that's all of the pathways and all of the enzymes and all the different things that you need to know. It might seem a little bit overwhelming. You can stop the video here and be done. The next several series of slides are drawn out like we do on the butcher paper. I've got it kind of drawn out with just a different way of looking at it, in a way of looking at it more with all the when you can see all the pathways at the same time. So like I said, like you don't have to, there should be a check mark there. You don't have to watch the next part of it. You can look through the slides on your own time. But I'm just going to go through them so that at least you have like one other way of looking at this. Okay, so this is regulation drawn out. And so the image that we're going to be looking at is this one right here. I've got it already drawn out. And this one doesn't have, this is only carbon intermediates. Okay, so none of the coenzymes are drawn. ATP is not drawn, well, except for this cute little ATP down here from ATP synthase. And some of the shuttles are also not here, but all of the major pathways are here. We have glycolysis running through here to the PDC to TCA. If you go up in this direction and out, you have gluconeogenesis. You have pentose phosphate pathway here, glycogen synthesis going on here. You have fatty acid activation and carnitine shuttle transport to beta oxidation. And then, you know, the acetyl-CoA needs to go into the TCA cycle. We have the citrate shuttle, where citrate shuttles out acetyl-CoA to be made into malonyl-CoA, which goes into fatty acid synthesis. So again, this is very, very basic drawing without all the coenzymes and intermediates, or without all the coenzymes and ATP, but only the carbon intermediates are shown here. All right, these are one version of what sometimes we refer to as the regulation tables. And these are just a good way to practice and organize the information in your head. So you have a high energy table, a low energy table, the ratios that are considered high and low energy, and then these would be the pathways that are upregulated and downregulated and high energy and low energy. These are tables that you can fill out as you're going through and studying. Okay, so the first one we have here is high ATP, and you'll notice that high ATP shuts down glycolysis at, shuts down glycolysis. This is just very broad overview. Shuts down glycolysis, shuts down the PDC, and it upregulates gluconeogenesis. Whereas low ATP, you can see upregulates PDC, upregulates TCA, upregulates glycolysis, and then downregulates gluconeogenesis. So you can see that these aren't even complete opposite as far as regulatory mechanisms go. Sometimes you get more. In this case, we get more pathways that are being turned on by low ATP than we're being turned off by them. High NADH will turn off PDC, the TCA cycle, and beta oxidation. And then low NADH will turn all of these on. PDC, TCA, and beta oxidation. High G6P, which means low G, reciprocally regulates gluconeogenesis, so turns on gluconeogenesis, turns off glycolysis. And by turns on and turns off, I really should be saying slows down and speeds up. Better, more flux through versus less flux through. Okay, and then the opposite is true. So when we have high glucose, which is a low energy signal, it means turn on glycolysis. We need to make lots of ATP versus low G6P shuts that one off, gluconeogenesis. All right, high acetyl-CoA. High acetyl-CoA, again, that is a product inhibition of the PDC, and it's also product inhibition of beta oxidation. High acetyl-CoA is a high energy signal, which means you need to stop making acetyl-CoA and start. You need to start storing the energy instead of using the energy. And then low acetyl-CoA, again, is the opposites here. High malonyl-CoA. So high malonyl-CoA is a high energy signal. It will shut down the carnitine shuttle so that the acyl carnitine is not moved, will not be moved into the matrix and therefore not broken down into beta oxidation. High malonyl-CoA also upregulates fatty acid synthesis. It's a substrate for it. And then the opposites are true. If you have low malonyl-CoA, that's a low energy signal. And that means that when you have low malonyl-CoA, low energy signal means you need to make ATP and stop storing the energy. So the acyl-CoA will be transported into the matrix, beta oxidation, TCA cycle, and then to the ETC to make energy. And by what we mean is make energy is make ATP. High citrate allosterically activates fatty acid synthesis. It's a allosteric activator. And then citrate, it inhibits that. So if you have low citrate, that means that TCA cycle is actually occurring over here and you're getting ATP and you need to make ATP instead of store the energy. Woof, okay, that's all that I have. Okay, breathe, meditate. You're going to be fine. I know you're going to do great. I'll see you in class.